Hi everyone, my name is Plugaro. I'm a second year medical student and today I'm gonna try my best to explain to you a very popular nowadays topic, the coronavirus. In this video, I will speak about the origin of a virus, its epidemiological aspects, about the means of transmission, its symptoms, structure, and finally, the infection process and the immune response. So without further ado, let's start. The name of a virus is SARS-CoV-2 and it's a beta coronavirus that belongs to a Coronaviridae family. Its name stands for Severe Acute Respiratory Syndrome Coronavirus 2. It's named so because it causes a severe acute respiratory syndrome, and 2 because there was a different coronavirus outbreak in 2003, but at a much smaller scale, and it was called SARS-CoV-1. The disease causes it's called COVID-19, which stands for Coronavirus Disease 2019. But I'm sure you all heard about that already. To better understand this disease, first, let's talk about its origin. The first case reported to World Health Organization was on the 31st of December 2019, and it all started in a city in Hubei province in China, and that city is Wuhan. It's thought that COVID-19 is a zoonosis disease, which means the virus that caused it originates from a non-human host. In this case, it's believed to be a bat, just like in the case of similar coronaviruses. But the bat virus can't directly infect us. In order to do so, it has mutated, infected another intermediate host, which we don't know which one is it, then mutated again and infected humans. The SARS-CoV-1 and MERS-CoV, which are very similar to SARS-CoV-2, also originated from bats, then infected dogs and raccoons in the case of SARS-CoV-1 and camels in the case of MERS-CoV. Um, in fact, viruses are so similar to the SARS-CoV-2 that it shares as much as 79.5% of SARS-CoV-1 and 50% similarities with the MERS-CoV and 96% with RATG13, which is the bat coronavirus. Also to be noted that both of the human viruses that I'm comparing it to have fatality rates of 10% for SARS-CoV-1 and 30% for MERS-CoV, which are much higher than of SARS-CoV-2. Now let's talk about the epidemiology of COVID-19 and find out what makes it such a threat. On the whole planet, as of 2nd of May 2020, there are 3.35 million people with confirmed COVID-19 diagnosis, from which one-third of all cases are in USA. By dividing the total number of deaths by the total number of cases, we estimate a fatality rate of 7.13% worldwide. Interesting fact is that the fatality rate of COVID-19 has almost doubled now compared to the fatality rate worldwide at the beginning of March. From this table, we can see that the highest fatality rate is in Italy and the lowest is in Germany and the poor country in Europe, Moldova. Why is it so, and why has fatality rate grown in general? Well, because the fatality rate highly depends on the speed of introducing restrictive measures and the amount of people that the state hospital can maintain with the necessary care. In Italy, the number of cases quickly boomed and the healthcare system was not prepared to handle that many cases simultaneously. So a lot of people didn't have the artificial mechanical ventilation they needed because all of them were already in use. So the fatality rate quickly grew. Other epidemiological aspects of a virus are the incubation period, the basic reproduction rate, and the serial interval. And I'm going to explain all of them one by one. The incubation period is the time it takes for the infectious pathogen, once infected a host, to develop symptoms. The incubation period for SARS-CoV-2 is about 5 days, ranging from 2 to 14 days, and some sources say that it is up to 24 days. The basic reproductive number, or the R0, is the expected number of cases generated from one case in a population susceptible to infection. For SARS-CoV-2, the R0 is estimated to be anywhere between 2 and 3.5. This means that one person infected with COVID-19 will usually infect three other persons during his disease in conditions where no special measures of protection are included. The serial interval is the time between successive cases in a chain of transmission. For SARS-CoV-2, is estimated to be between 4 and 8 days, averaging at 6. With that out of the way, let's now discuss the epidemiological differences between SARS-CoV-2 and the influenza virus, also known as the common flu. As we can see, the influenza virus has a smaller R0, averaging at 1.4 to 1.6 and a lower SI, which makes it infect less people from one case, but almost twice as fast. In the following diagrams, I will round up some numbers for simplicity reasons. The main point of me showing it to you, dear viewer, is to understand the concept. So for SARS-CoV-2, the diagram to the left, we will consider R0 to be equal to 3 and SI equal to 6. This means that one infected person will infect other 3 persons in an interval of 6 days, and that 3 persons will infect themselves in a period of 6 days, 3 persons each. So that will be 9 persons. In total, we have 1 point, plus 3 plus 9, and that will be 13 cases in 12 days. A mathematical representation of this sum would be R0 to the power of 0, which represents the first infected person, plus R0 to the power of 1, which represents the recently infected people from the first case, plus R0 to the power of 2, which represents the people infected by the second wave of infected persons, and it can go like this indefinitely. Each generation will add up to the power of R0 plus 1. On the right diagram, we can see the prognosis of influenza virus spread. We'll round up the 0 to 2, even though the most common value should be around 1.4 and 1.6, and the SI to 4 days. During the same period of 12 days, influenza virus patients would have time to infect 3 generations, instead of 2 like in SARS-CoV-2 case. Because of shorter SI, we get n, the number of new generations, equals to 12 days with an interval divided by SI, which is 4, and that equals to 3. So the n is equal to 3. The total number of cases would be 2 at the power of 0, which is the first infected, plus 2 at the power of 1, which represents the first new generation, plus 2 at the power of 2, which represents the second new generation, and plus the 2 at the power of 3, which represents the third new generation. So we get a total of 15 infected, greater than SARS-CoV-2 by 2 units, but keep in mind that we rounded the numbers for simplicity's sake. The main point of these diagrams was to illustrate the differences between SARS-CoV-2 and influenza, which are the number of cases infected from one case and the time it takes from the onset of symptoms on one individual to the onset of another individual in a chain of infection. And we learned that COVID spreads to more people from one case but slower, while influenza does it faster but to a lesser extent. In total, we scored a similar number of new cases in the same period of time. So why is it then that the SARS-CoV-2 is so threatening that almost every country shut down their borders? Well, it is so because unlike for seasonal influenza, there is no vaccine for SARS-CoV-2, and that there are so many complications in the disease throughout patients that there will be not enough space in the hospitals to maintain all patients if we would let the virus run wild and not take any actions. Now let's discuss about the means of transmission of this virus. First means of transmission is through respiratory droplets. These are small liquid molecules in which a virus is present. These droplets come from a cough or a sneeze and can aerosolize in air for up to 3 hours and travel distances of tens of meters. This means that you should always wear a mask and don't think that if there are no people around you in the supermarket, you are free to get your mask off. The second way of getting infected is through touching contaminated surfaces and then touching your eyes or mouth without washing your hands. It's helpful to know the time which the virus can survive on different surfaces since we touch them on a daily basis. On plastic and stainless steel, the virus can survive for a relatively long period of time, which is 3 days. On copper, it can survive 4 hours, and on cardboard up to 1 day. So next time you get your Amazon order, you can just leave a box for 1 day if you're afraid of touching it. The last way of transmission is through the fecal-oral route. It is thought to be possible, but there are no registered cases of infection through this specific way. Now that we know the means of transmission, let's talk about the symptoms. The most known ones are dry cough, but sometimes in 30% of cases it can be wet cough, and also fever. This virus can infect any cell that has a specific protein which is called ACE2 receptor. So the cells that make up the respiratory tract, including olfactory cells and even the cells from the tongue involved in the feeling of taste. This can lead to hyposmia, decreased smell sensitivity, or anosmia, the total lack of smell, dysgeusia, the disturbance in the feeling of taste, and also sore throat. More severe respiratory symptoms include pneumonia, acute respiratory distress syndrome, systemic inflammatory response syndrome, and multisystem organ failure. Because there are ACE2 receptors in the heart, kidneys, and gastrointestinal tract, SARS-CoV-2 can also cause cardiomyopathy, decreased renal function, and even diarrhea. I'm still mentioning this ACE2 receptor, but now I'm going to explain the structure of a virus and what part of it binds to our cell receptor. So the SARS-CoV-2 is made up of five main components. The spike proteins, abbreviated as S protein, its membrane, which is made of M proteins, the E protein, genetic material, which can be either DNA or RNA, and you know that it can be a single-stranded SS or double-stranded DS. Well, in our case, to be specific, is positive single-stranded RNA. The word positive in this context means that this RNA is ready to be translated into proteins, so it's 5' to 3'. If it were a negative sense, it would mean that an RNA-dependent RNA polymerase would have to replicate it firstly, and the newly synthesized RNA would be positive sense, and only then it would be able to be translated. The last major structural component is the nucleocapsid, which is made of N proteins and helps protect the RNA. Now back to S protein and why it is so important. The answer lies in its structure. The spike protein is made up of two parts, VS1 and VS2. VS1 is that important part which helps the virus bind to ACE2 receptors in our cells, while the ACE2 proteins help the virus fuse in them. We finally made it to the last and most interesting part, the infection process and the immunological aspects of SARS-CoV-2. This topic is very vast and complicated. I'm gonna present it as known as of today, but new details might emerge later. So, how does the infection get into our body? We discussed this in the transmission section, and it has to be from the virus particles in the droplets, from contaminated surfaces, or fecal oral route. In this diagram, our supposed patient, John, inhales the aerosolized droplets into his upper respiratory tract, then into his lower respiratory tract, and finally, the virus gets into the alveoli. Do you remember from what type of cells the alveoli are made of? Right, it's made from pneumocyte type 1, which secures the gas exchange, and pneumocyte type 2, which produces surfactant. Well, this cell and the respiratory pathway cells in general have this receptor ACE2, which the virus uses to enter the cell. Now let's take a look at the intracellular level. What does this virus do? For our next diagram, a pneumocyte type 2 will be used as the host cell, since it has that specific receptor. First of all, the virus binds to the ACE2 receptor found on the surface of the cell and enters the cell via endocytosis. Once in the cell, it frees its positive single-stranded RNA. Once it's free, two things happen to it. Firstly, it gets translated by a free cytosolic ribosome or one paired with a raw cytoplasmatic reticulum. While translating an RNA, what does the ribosome make? Right, it makes immature proteins. From the SARS-CoV-2 RNA, four structural proteins are being made, the ones we discussed before in the video. Also to be known is that the first form that these proteins come in after translation is not their final form, so they are being cut by specific proteases, which are enzymes that eliminate some parts of the newly formed proteins in order to help them mature. This RNA codes for 16 non-structural proteins, NACPs, which helps evade the immune response and replicate the virus. One major non-structural protein is the NACP12, which is a RNA-dependent RNA polymerase, which uses RNA to make copy of it with reverse sense. The second thing that happens to RNA is it gets multiplied by NACP12 and its quantity in the cell quickly rises and is being again and again translated to make even more viral proteins. Also, it gets combined with the N proteins, forming the nucleocapsid with a positive single-stranded RNA inside. The nucleocapsid, together with S, E, and M proteins, go through the Golgi apparatus and undergo a series of conformational changes and are being assembled together to form a new copy of a virus. Then the vesicle of a newly formed virus fuses with a cellular membrane, and the virus is free from this cell and ready to infect other cells. This process repeats over and over again until all of the energetic resources of a cell exhaust in replicating the virus and the cell dies. Now, when that is out of the way, let's talk about the immunological aspect. Don't be scared of this diagram. We will go slowly through it, and at the end of you will understand the pathology of a disease. What is it that causes those symptoms mentioned before? And generally, you will not be afraid of this virus anymore. So in order to navigate more easily, you should know that the primary path is shown with orange. Function of a certain cytokines and secondary information shall be colored in deep green, and important path components and symptoms will be colored in a pinky red. In the last diagram, we used a pneumocyte type 2 cell, which alongside of pneumocyte type 1 makes up the alveoli. Now let's zoom out of a pneumocyte to a view of alveoli, which are basically tiny sacs of air in which gas exchange occurs at the respiratory membrane, which is very thin. The respiratory membrane is made of alveolar wall, the capillary wall, and their basement membranes. So now let's repeat. What is an alveoli made of? It's pretty simple actually. It's made up of two types of cell. The type 1 pneumocyte, which makes the gas exchange of O2 and CO2 between inhaled air and alveoli capillaries, and the type 2 pneumocyte, which make the surfactant. And what is surfactant for? Well, it lowers the superficial tension between the little quantity of water present on the surface of alveoli and the air. If were not for surfactant, the alveoli would collapse and we would not be able to breathe. So we showed how pneumocyte type 2 got infected. Let's say that the virus started spreading and killing off the cells in the way. Once it's floating around, a professional antigen presenting cell, such as a macrophage, the dendritic cell in our example, will sense its presence by attaching to the spike S protein with a toll-like receptor 4 or by attaching to a viral RNA with a toll-like receptor 3. After the attachment, a series of reactions happen within the cell and make the dendritic cell produce tumor necrosis factor beta and interferon alpha, from which interferon alpha, by combining with interferon alpha receptors, activates through JAK-STAT pathway the STAT1 and STAT2, which will activate the transcription of specific genes in the nucleus that respond for many cytokines such as tumor necrosis factor alpha, interleukin-6, transforming growth factor beta, interferon gamma, and others. They are very important in the immune response because tumor necrosis factor alpha affects the hypothalamus and induces fever and the loss of appetite. Fever is induced so a viral peptide would lose their tertiary structural integrity and function, and that's a very important symptom. Interleukin-6 and transforming growth factor beta help differentiate the CD4 positive cells into T helper 17, which would produce interleukin-17 and, in the end, recruit neutrophils to the site of inflammation. In a casual viral infection, this would be very helpful in restraining the replication of a virus and would decrease the severity of the disease. But in this case, the cytokines are produced in excess and cause a lot of harm to the organism, leading to hyperinflammation and to the acute respiratory distress syndrome. So what happens in inflammation? Mainly, what happens is that macrophages sense DAMPs, damage-associated molecular patterns, that come from the dead cells, and PAMPs, pathogen-associated molecular patterns, which are part of virus proteins, like when I told you about toll-like receptor 4 recognizing the S protein, and they secrete vasoactive amines like histamine and serotonin, and cause the plasma liquid and plasma proteins to start leaking into the tissue. This process decreases the normal diffusion of oxygen and CO2, which will then lead to hypoxia and hypercapnia, and also acidosis. And what will hypoxia and hypercapnia lead to? It will lead to a shortness of breath, which is also a symptom. Not only that the inflammation affects the diffusion, but also the death of pneumocyte type 1 and type 2 because of aggressive immune response where neutrophils do a lot of damage to the healthy tissue, and from that the death by exhaustion from virus leads to decreased gas exchange once again. Why? Because type 1 pneumocytes were responsible for the exchange of gases. And if their number is getting lower and lower, so does and the gas exchange. Also, by the death of type 2 pneumocytes, the surfactant production drops low. There is less surfactant on the internal surface of the alveoli, and breathing becomes harder and harder. This will eventually lead to alveoli collapsing, and once again, hypoxia and hypercapnia. And we already know what that leads to. They lead to shortness of breath. Meanwhile, another professional antigen-presenting cell, such as a macrophage, that already presents the viral peptide on the MHC type 2, will activate the T helper cell and secrete IL-12, so it will differentiate into T helper 1. The interferon gamma discussed earlier will also help in the differentiation of T helper 1 and also will rise the activity of the macrophages. The T helper 1 cell will start secreting interleukin 2, which will activate cytotoxic T cells. We'll also secrete interferon gamma, which will, as I said previously, increase the activity of macrophages. And another function of it is increasing the MHC class I expression. T helper or other immunocompetent cells will be activated by tumor necrosis factor alpha through a much important NF-kappa B pathway. It will lead to a secretion of a bunch of cytokines, such as interleukin-1, which has pro-inflammatory functions, interleukin-6, which helps activate the T follicular helper, which will produce interleukin-21, which in the end will activate and help cytotoxic T cells and natural killer cells to proliferate. Also, interleukin-8 will be produced, which will increase the chemotaxis and the phagocytosis activity of neutrophils. This will once again, as I said, cause healthy tissue casualties, the death of type 1 and type 2 pneumocytes. Another cytokine produced is tumor necrosis factor beta, which will increase the secretion of chemokines and expression of MHC in secondary lymphoid organs. And last but not least, the MACP1, which will recruit monocytes, memory T cells, that will eventually live up to 4 years after infection. And it will also recruit dendritic cells, and it will recruit all of them to the site of inflammation. In essence, all of this hypersecretion of pro-inflammatory cytokines, spreading of a virus through the body to other organs that have ACE2 receptors, will lead to systemic inflammatory response syndrome, which is basically an inflammatory state affecting the whole body. Eventually, if a virus isn't eradicated in time, this will lead to multiple system organ failure and death. But we know that the mortality rate is only 7%. The rest do survive it. So how does the organism get rid of SARS-CoV-2? First, a T helper with a T cell receptor with affinity towards usually the spike protein will find and activate the B cell through T cell dependent B cell activation. B cell will start producing immunoglobulins as the first response line, but later on in the infection, the more efficient G immunoglobulins will be seeing production. In the end, these immunoglobulins will eradicate the SARS-CoV-2 from the organism for good. I hope this video was helpful for you and that you understood where does this virus come from, how does it spread, its symptoms, and pathology, and that you should not be scared of it and know how to protect yourself from it. For today, I was your host, Bulgarodan.